Hi, everybody. Can the people online also hear us? Can I have a thumbs up from somebody to indicate that things are going well? Yeah, cool. Oh, thumbs up. Thank you. We should do this more often. Okay, it is my pleasure to introduce Dr. Robert Wright, who is a pediatrician and medical toxicologist and environmental epidemiologist. And he told me even secretly also trained in genetics. He works at the ICANN School of Medicine at Mount Sinai as an international recognized expert on the health effects of chemicals and environmental exposures. He co-founded the Institute for Exposomic Research and chairs the Department of Environmental Medicine and Public Health. Career highlights are that his education, he received his MD from the University of Michigan and his Master's of Public Health from the Harvard School of Public Health, where he also completed multiple fellowships, including those in medical toxicology and environmental epidemiology. Focus is on exposomics, and that's why we invited you here for this topic in the grant rounds. Is one of his major focus and the study of health relevant human exposures throughout a lifetime. His research shows that environmental factors and chemical mixtures interact with a person's genetics to affect health. At the Institute for Exposomic Research, where he was the founding co-director, is the founding co-director. They also study the impact of climate change and how that influences health outcomes besides the exposome in more general. He leads several, he's principal investigator of several cohort studies, very long-running studies. For example, the Progress study in Mexico City, which looks at the impact of environment and obesity growth and stress and follows kids over time. And a nice fact about Dr. Wright is that he also has a blog where he offers unique perspectives on environmental health research and exposomics. So you can follow him there, and he has won numerous awards. And without further ado, I would like to give the floor to Dr. Wright. Thank you.
Thank you, Fed, and thank you for doing that plug, because I always shamelessly plug my blog, my slides. So if you just Google Exposome blog, I think you'll find it. I think it is, it may be still the only blog out there. Oh, where's the mouse? Do I need the mouse? Ah, yeah. Maybe use this.
No. Oops, or maybe just forward. Oh, so not. Oh,
okay. So I'm going to assume that a lot of you are toxicologists, and you've all heard the term from Paracelsus that the dose makes the poison. So I'm going to argue that there's actually another set of rules, and that is the context also makes the poison, and that exposomics is actually key to understanding context. So I'm going to present some ideas for Human Exposome Map, and I am naturally conflict averse. So I'm going to create a disclaimer that there is no right or wrong way to conduct a human exposome study. So this is just a proposal. Some of this may actually happen. A lot of it probably won't. But it is compatible with all other proposals. And the main goal is really just to spark some discussion. And hopefully we'll get started soon, because I think the only way to learn about the human exposome is to actually try to do the project. And I think we spend far too much time, I think, defining the exposome. I would much rather we just get started, because I think we'll learn a lot more from the process of doing. So here are some key questions for human exposome projects. So biggest question is, how are you going to convince government or industry that this is important? I'm going to try to address all of these in the talk. How will it help the economy? That's probably the most important question if you want to get it funded. I used to be someone in the camp that, well, that shouldn't matter. It's just about, what's best for the public. But in reality, how it's going to help the economy is probably going to be the deciding factor. what would we do with the information if we had a human exposome project? So what's its value as a research tool or a clinical tool or a public health tool? So we have to think about that and have that an answer to that question in advance. How will it start? How will it end? That's probably the easiest part. I think you can start whenever we want to start it, and we can decide how to start it. I don't think it'll ever end because our exposome is constantly changing. It would just have to be updated. And then the question I think is going to come up because research funding is limited and we're all competing for the same pie. Why shouldn't we just continue to focus on genomics and multiomics and then first complete the Human Genome Project and then move on to exposomics? And I think we're going to hear this question. But, for those of you who like Disney and Lewis Carroll, I think we just need to begin. Begin at the beginning, the king said very gravely, and go on till you come to the end. And that's advice from Alice in Wonderland. So I always feel like I have to make this distinction between geographical and biological maps. So when we think about maps in biology, we think about biological maps, the Human Genome Project, the epigenomic map, the metabolomic map. So where do these different biomarkers divide in cells or divide in different tissues? I'm really talking about a geographical map, a geospatial temporal map. My argument is the exposome is actually geographical and it changes by calendar year, as well as biological. So it's integrated into what could be integrated into a biological map. And a biological map or atlas could be incorporated into a geospatial map. There's nothing stopping us from doing that. In fact, if you look at genomics, they actually use reference exposomes from different parts of the world. They're actually thinking geographically. So geographical maps are natural and they're part of biology. And we don't really have to choose. If we had a biological map or a biological atlas of the human exposome, we could integrate it into the geographical map. So why do we need a geospatial exposome map? My argument is the exposome map is the key to understanding context and how it plays a role in toxicology and in human health. But also, perhaps most importantly, I think we can use it to go back in time, that we can actually reconstruct our past with maps. So what is context? I'm going to define what I mean by context. It's the who, what, when, and where of an observation or an experiment. Context could be geography. Our exposome is very different here in Baltimore than it is in upstate Pennsylvania. It's social cultural, which depends on how you interact with people and your diet, among other things, as well as your family. It's political, and it's the economic setting in which the research takes place. You notice I skip past politics really quickly. So chemicals in cells, like mixtures, or a type of context. Genetics, gene-environment interaction, is another type of context. And all those things can affect the results of an experiment or an observation. Dose itself is considered, or you could consider it a context, particularly nonlinear dose-response curves. The mechanisms at low doses may be very different than the mechanisms at high doses. We see that all the time. For lead poisoning, the mechanisms at low doses had more to do with neurotransmitters. The mechanisms at higher doses had more to do with inflammation. So in this context, exposomics can be thought of as the means to better study context in toxicology. And here's some examples. So if you did an experiment using this cage and you compared it to animals that you did the exact same interventions, but use these cages, these social enrichment cages, would you expect to get the exact same result? You'll probably get very different results because social environment will modify the context of lead poisoning. In fact, Tom Yarty did some experiments exactly like that here about 20 years ago. And then there's genetic backgrounds. As I said, gene-environment interaction is a type of context. So I study neurodevelopment. So if environment has no impact on plasticity, you would see parallel lines. If it has impact on toxicity, you'll see some non-parallel lines. And if that highly variable impact of environment varies in different contexts, it could be because there's a genotype by environment interaction. And nutrition can be a context. So this is a map of Chicago where there's food deserts and where life expectancy is different. And you overlap the food deserts with the life expectancy, you probably see a lot of overlap. This is really why I think geographical maps can be so helpful, because you can actually look at where things are co-located, just as we do with biological maps, where things are co-located in cells or tissues. And then life stages of context. The guy in the upper right is David Barker. So this, when I was first starting out, this idea was actually called the Barker hypothesis. It's now called the developmental origins of health and disease. It was because he was really the first person to describe the idea that if you get exposed to something very, very early in life, it may actually predict your health 20, 30, 40, 50, 60, 70 years later. Maybe Alzheimer's disease has its origins in childhood lead poisoning, as an example. So life is also a trajectory, and early life events are very, very important. However, the problem is studying early life is really, really hard because we only exist in the present. Or if we want to do a study today and we want to know whether or not childhood lead poisoning causes Alzheimer's disease, we'd have to start today with children, follow them, measure their lead, 80 years later, see if they develop Alzheimer's disease. Well, maybe our great-grandchildren can do that because we'll all be dead by the time that study is done. So unless we can reconstruct the past, this is going to be a really, really hard study to conduct. And it's important because life is a trajectory. So if you get offset in that trajectory early in life, it causes a much bigger gap. Well, you don't see that. It causes a much bigger gap. This doesn't work either. Oh. It causes a much bigger gap later in life than if you start with that trajectory offset here. So if I'm traveling between New York City and San Francisco and I'm off by 1 degree, I'll be about 50 miles away from San Francisco. If I start in Salt Lake City and I'm off by 1 degree, I'll only be 3 or 4 miles away from San Francisco. So it makes a big difference when these offsets and trajectories happen. And in fact, in our research currently, this is a black box. We're studying Alzheimer's disease here. We think this might be important, but we don't know anything about it. Maybe we could learn something about it if we had an exposome map, because I think with an exposome map, it may be possible to go back in time and reconstruct the past. And I'll explain how I think that could be done. So there's a lot of barriers to exposomics. 2 aspects appear to be the most difficult. We talk about the first one all the time, measuring all the environment, the everything, everywhere, all at once idea. So whether it's social, chemical, physical, biological, we're measuring everything. But we tend to do that cross-sectionally. Remember, the exposome is all your environment from conception to death. That's the harder one. We don't really talk a lot about that. But that's actually the key. I think that's the game-changing part of exposomics, if we could go back in time. Because our health today is a function of our past exposome, not our exposome today. And in fact, Francis Collins, in his exit interview from NIH when he retired in 2021, actually said this: your likelihood of having a certain lifespan depends heavily on the zip code where you were born. And that is a reflection of the environmental exposure, socioeconomic, social determinants of health, etc. So even the world's probably most famous geneticist concedes that early life environment is important, which should tell you something about how important that is. But we have a really difficult time reconstructing that. So now I'm going to go into an analogy. So I've told a couple of people that I was once a literature major, and I'm a big fan of magical realism as a genre. And so my favorite author is Jorge Luis Borges, and he wrote a short story called Funes Memorias. And this is about Ireneo Funes. He was a man who had a perfect memory, and how remembering everything, everywhere, all at once, slowly drove him mad. So these are some excerpts from the short story. Funes remembered not only every leaf of every tree and every patch of forest, but every time he had perceived or imagined that leaf. It irritated him that the dog of 3:14 in the afternoon, seen in profile, should be indicated by the same noun as the dog of 3:15, seen frontally. To him, each memory of the dog was unique, not just the dog itself. Funes had no insights on life, only facts that he could recite in minute detail. What Borges was actually criticizing was a philosophical field called nominalism, which contrasted with a more common field called realism. In realism, which was actually described by Aristotle, it's a philosophy in which the world is defined by particulars and universals. Particulars share characteristics of universals. Cody is a dog, Frodo is a dog, Brody and Kodo are particulars, dog is a universal. In nominalism, there are no universals. There's only particulars. For example, there is no good or evil. Each behavior and action is unique. It's very Machiavellian. Cody is Cody, Frodo is Frodo, and there is no such thing as a dog. And taken to its extreme, Cody this morning is different from Cody who's playing with Frodo. These are my dogs. What Borges was doing, he's criticizing nominalist thinking as preventing understanding. So in some ways, we do this with omics. We just catalog lots and lots of stuff, and we think knowing everything has value, when in fact, most of what we measure is actually useless. There are 4 billion base pairs in all of us. 99.9% of all those base pairs are the same in you and me. The SNPs and the variants are actually a very small percentage. So why do we need to measure all that stuff? We spend a lot of energy measuring things that don't vary. And exposomics is in danger of doing that too, unless we have a plan. So the exposome, as Christopher Rao defined it, is composed of every exposure to which an individual is subjected from conception to death. It includes internal processes, external exposures, social, economic, psychological influences, and it's overlaid on exposure timing. So life stage matters, whether you get exposed as an infant, as an adolescent, as a young adult, or as an old adult, will have a different effect. My argument is that exposomics could become a nominalist science by trying to measure everything, everywhere, all at once, across time, without a plan for how to use that information. So I'm going to propose a plan. Again, as a disclaimer, this isn't the only possible plan, it's just a plan. Because I worry that, like Funes, we're cataloging the exposome instead of understanding the best ways to leverage it. So I propose that we could redirect exposomics towards discovering the past, because I think reconstructing the past would actually be the game changer that exposomics really needs to be adopted by other sciences. Changing the goal to imputing the important components rather than measuring everything makes this possible. And I'll explain what I mean by that. Also, AI makes this possible. So maps, by that I mean geospatial temporal maps, can anchor the physical, chemical, social, and biological exposomes together in a single platform, so that we can understand the role of geography and time, but also how all of these different environmental exposures vary across time and space. And it could be a good starting point. And in fact, genomics has been doing this. So how do we apply lessons from genomics to exposomes? Well, chromosomes are the genome's geography. Genome maps show that variants nearer each other on chromosomes tended to be inherited together. Regions of linked variants are known as haplotypes. So this led to the creation of what was called the hap map, which took advantage of how single nucleotide polymorphisms, or SNPs, and other variants were organized geospatially to efficiently conduct genome-wide association studies. So GWAS studies actually use this principle of geography. Genomics actually exploited geographical correlation in the genome in order to accelerate research. Genomic maps went from simple two-dimensional maps to three-dimensional ones that now actually are used to estimate conformation to predict transcription. So we started with simple maps and we grew to more complex maps. I think exposomics can learn some of the lessons that genomics learned by applying geography. So how are haplotypes used in GWAS? So the genome map revealed that there are haplotypes, and that is that SNPs tended to be inherited together if they were near each other. So if you have SNPs in a gene, big A, little b, big C, big D, and then that corresponds to little a, big B, C, D, these tend to be inherited as a block. So if you know that you have big A, you also know that you have little b, because they get inherited together. It's because of the correlation of these SNPs. And then these could be used, individual SNPs could be used as tagging SNPs. So if you know one of them, you know the rest of them. That's how hap maps work and haplotypes work. Maybe there's something like that going on in our exposome. It won't be exactly the same, but conceptually, there's a correlation pattern. There's an exposomic correlation architecture that exists. In fact, inherently, we know that if you know enough about me, you can probably predict a fair amount of my exposome, and I'll give some examples of that. But associations between these tagging SNPs and health outcomes or phenotypes represent an association with chromosomal regions, not necessarily directly, or probably isn't directly causative, but you now have candidates that you can zero on. So you take this information of 4 billion SNPs and you narrow it down to a smaller subset of SNPs. And today, reference genomes are used to impute unmeasured genotypes after a subset of SNPs are measured. So now we know that we have these reference genomes, which are thousands of people. They're not just single people, but they're thousands of people, and they're based in part on geography. And we use that to impute SNPs. And we can do this. So they call them haplotype tag SNPs. So just for the sake of having a nomenclature, I'm going to call these exposome tags. So these are environmental exposures at the individual level that correlate with other exposures. If we could better understand these, if we understood the exposome architecture. Exposome measures clearly have a correlation structure, even across categories. Social variables tend to go with chemical exposures. Lead poisoning is more common if you're poor. So there are these correlation structures that we know already. A human exposome map could be the linchpin for exposome imputation by giving a better understanding of these tagging exposures and how to use them in research. So it would look something like this. So it would be everything, everywhere, all at once across time. So a lot of this data already exists. We don't have to reinvent the wheel. There are external environmental maps of the United States and other parts all over the world that already exist. These maps for air pollution, climate and weather variables, healthy food access, allergens. There are area deprivation indices that you can get from the census data that can actually tell you something about your neighborhood. There are walkability scales. All of these maps already exist, but they're in separated silos. If we're going to do this, we can link them at the individual level to people based on where they live. And as Francis Collins pointed out, where you live is a very big predictor of your health. We could even do this for biomarkers. Blood lead does this all the time. The CDC actually does this. They create maps of lead exposure risks for children based on their screening data for blood lead that gets reported to them. But we don't have this for other chemicals, but we have data from other studies, relatively large studies like All of Us, ECHO, UK Biobank, Framingham Heart Study. We could start to map those biomarkers. And if we had residential histories and timelines, we might even be able to go back in time. If we know how far back the maps go, we can at least go that far back, and then we can start to assign exposures. So geographical and temporal mapping places exposure assays, social environment, climate, air pollution, physical environment, all into a common platform, which allows us to assess their correlation structure. And we could even start by collating existing data. And remember, exposome maps are temporal as well as geospatial. Air pollution, climate models, for example, go back in time. Everything on a map can reflect calendar dates in place. And chemical biomarkers, if we had them, could even go back in time if we knew the year in which they were collected and measure them. And we could, if we had all this information, create a life course geospatial exposome map. And in this example, if we map exposure biomarkers, each person is like an EPA air monitor, so we can use them to calibrate the map. So if we have this data, we can actually, and we have this address, we can actually treat it the way we treat air pollution modeling. And then once we have that, we could start to look at the correlation over space and time. And in this figure, red is the missing exposome data, green is the measured exposome data. And if we know the correlation structure, we can start to impute the missing data with the data that we're actually able to collect. And this is the way most air pollution studies actually work, because there are days in which they're cloudy, and you're going to have to impute what your missing data are, because you have missing data if you're using satellite data and there's clouds. People do this all the time. You're probably doing this yourself in your cohort studies, where you're trying to impute the missing data. So with a temporal geospatial map of everything, we can determine the exposome's correlation structure and then use that information when we do a study to impute the parts of the exposome that are missing and the parts that happened in the past, if our maps go back far enough in time. That's going to be the hard part, going back to 1950. So people who have Alzheimer's disease today, they were born in the 1940s and 50s largely. So satellites didn't exist back then. So there's going to have to be other ways to do that. But I think with AI and data mining, there are probably going to be ways to get back at that level. There's going to be greater error bars the farther back you go in time. However, it's a better estimate than what we currently have, which is nothing. And I think using machine learning and multiple imputation, this is actually possible now. And so here's some examples. These are what I would think are important exposome tag measures. And I picked these because it's relatively simple and I can give you an example of them. So here are some that I think tag other measures. So where you live, your home location, that's going to tag traffic pollutants. If you're rural, it might tag water contaminants and higher risk for that. Life stage might depend on some of your activities, may tag whether you play or in sports or you're more sedentary, and what your diet might be. An infant's diet is very different than a teenager's diet, which is different than an adult's diet. And then income, products, occupation, all these things are probably important tags. In fact, I think if I only know 1 and 2, I can make pretty educated guesses about your exposome. If I know 3 through 6, I can make even better guesses about your exposome. And one day, if we know 7 and 8, we can actually get even better at predicting your exposome. And I think geography plays a big role in this. I'm going to give an example. So I'm going to give you my life history for address and life stage and think of them as exposome tags. So I'm going to do that for my life stage. This is my own private exposome. That's stolen from a River Phoenix movie from the 1990s called My Own Private Idaho. It's a good movie. So I was born in 1963 in Okinawa. So I was born on a military base. My father's in the military, my mother's Okinawan. For the first 3 years of my life, I lived across the street from the entrance to Kadena Air Force Base. If you've ever been in Okinawa, Kadena is the largest Air Force base there. And that was in the 1960s. This was during the Vietnam War. A lot of Agent Orange was going in and out of Okinawa. So I may have been exposed to Agent Orange. Who knows? I lived across the street. There was a lot of spraying on the base because it wasn't appreciated how toxic that might be. And you can find that from newspaper articles from the 1960s. So in 1966, I moved to downriver Detroit. I lived in an area very close to the Ford Rouge plant. So it was very urban. And it had probably a lot of air pollution because it was the 1970s. EPA didn't exist until I believe 1973 or 72. And so there wasn't a lot of regulation factors. And actually the most polluted zip code in the country is 48217, which is actually next door to my zip code. I skipped over 89 and 93, but I lived in Chicago for those 3 years, but it got to be kind of crowded on the side. So from 1993 to 2012, I lived in suburban Massachusetts in a very rural area, but it was a suburb, lots of green space, probably very low air pollution, a lot of pollen. I used to have a lot of allergies when I lived there. And then since 2012, I've been living in Westchester County, which is a suburb, but it's pretty crowded compared to where I used to live. So there's probably a lot more traffic pollution. So having more information would actually refine the map, but even with this, you could probably make some inferences about it. So I lived on a military base the first 3 years of my life. I might have had some chemical exposures because of that. My early childhood, I lived as a teenager. Up until I was a teenager, I lived in an older house, probably lower middle class. Probably shouldn't say lower SES. My parents would have been upset by my saying that. Air pollution was probably very high, and metals and chemicals from the auto industry were probably being thrown around. It was the 1970s. Young adult, I was in Chicago, urban, middle class physician, probably upper middle class by the time I started making a little bit of money. Low crime area, sparsely populated, high power, lots of green space, low air pollution. And now suburban, still low crime, more densely populated, moderate green space, more traffic exposure. So you have a fair sense of what my exposome has been like at different life stages. So I probably have a more variable exposome than a lot of people. I grant you that. but there's a lot of information that can be gathered just by knowing my address in the life stage. And that's just 2 of the exposome tags that I proposed. Much of that information we missed if only my current address is used. That's why I think we have to have address history. And how often have you ever been asked address history by your physician? My guess is 0. How often have you, people that do health studies in humans, ask for address history? I never have. Well, that's good that you're doing that, because we really want to know life stage. We want to know when you were exposed as well as what you were exposed to. Right now, these maps are kind of primitive, but if we had better maps, we could better estimate my past exposures. And I don't think it would be that hard. In fact, as I said, there's a precedence for mapping chemical biomarkers. The CDC already does it. The methods for this already exist. They get all the lead screening data across the country. It gets funneled to them, and they create models for who's at high risk based on the lead screening of your neighbors. So you don't have to actually be screened in order to fit into this model. Ultimately, if we had a map of everything, whether it's crime or SES or lead or whatever it is you want to study, if you linked it to something like autism spectrum disorders, you could start to see the co-location of all these different environmental exposures and different health outcomes. And you can see if they co-locate. That doesn't mean that that's where you should stop, but it's a signal that maybe deserves further study. I recognize that this is ecological at some level, but it's clearly something that's happening, that we could actually narrow down the greater exposome and the greater phenome to drive more important questions that the map could actually reveal need to be studied. And chemical mixtures could even be represented on such a map. You could look at different types of exposures and you can overlay them geographically, and you can see where they relate to different health outcomes as well, which would also be on the maps. And that might be pilot data to justify whether you want to do a particular study. If you're doing a study in a particular geographical location, and we all do by definition, and you have to do it in some geographical location, you could look at what's common in those geographical locations to guide what is the important environmental exposures and the other contextual measurements that you need to have in order to do the study in a way that's relevant to that population. So it could be built initially, this human exposome map with existing data designed to capture the past and present. We already have lots of data resources. NASA has satellite data going back about 25 years. We have census data going back even farther. NOAA has data, USGS, US Geological Systems, EPA, all these different governmental organizations have been collecting data that can be mapped going back decades. And geospatial statistics, thanks to all the air pollution, thank you, Joel Schwartz, there's a lot of methodology that's already been done on geospatial statistics so that we can actually look at the geospatial variation of exposures and health. And we can add to these mapping biomarkers from existing cohorts and going forward, trying to map all these. We have a data sharing requirement now at NIH. Maybe we could start to create data repositories that include address and maps so that it could actually start to build on itself without having to reinvent the wheel all the time. And we could even target older biobanks that have address data, like the National Perinatal Collaborative, which is which is a study done in the 1960s of children's health, and also has a biobank. Actually, I envision that this human exposome map could at least start as a data architecture that captures, collates, and harmonizes exposome data across life stage and geography. And actually, the idea that this is a data architecture is actually Marilyn Miranda, who's a very well-known mapper, data scientist, and statistician, who's actually the chancellor of the University of Illinois, Chicago, and a good friend who had lots of conversations with on this. And I think she would actually be the perfect person to run a human exposome map project like this. But I believe temporal spatial maps are the foundation by which we can impute the past. And we could still incorporate a biological atlas. And in fact, I think the biological atlas would be even more informative if we could see how it varies over time and space. So I try to make the argument today, and hopefully I convince you that a human exposome map that can be used to reconstruct the past is really the Rosetta Stone, not just exposomics, but developmental origins of health and disease, gene-environment interactions, and health disparities. So if we have this map, all of these case control studies, let's say of autism, that have been conducted, if they have address, we could reconstruct what their environmental exposures might have been during pregnancy, which is the life stage that's most important for autism research. And we could do that without having to re-enroll anybody. So think about the different ways such a map might actually be very valuable as a research tool. And I think this map could be the grand scientific challenge of the 21st century if we can use it to go back in time. So I recognize I'm a toxicologist. I recognize that lots of toxicologists in the room. Maps don't help me. The dose makes the poison. I think it's the context, but it can help you, is my argument. It could be a resource for mixtures. So if you have a population in Baltimore and you want to know what mixtures to study, you could pull it out of thin air, which is what we've been forced to do over the last 40 years. Or if you had a map, you could say, oh, the population that I'm studying lives here. What else is here? Maybe there's a Superfund toxic waste site. There's going to be the chemicals listed in there. This sort of map could actually direct you to what's most relevant to the population that you're studying, as well as the social context and potentially even the nutritional context. Also, the relative doses might give you some information. Instead of picking up doses out of thin air, you might see that there's 10 times more lead than cadmium. Therefore, whatever experiment I do has to keep that ratio in mind. It could be a resource for social chemical interactions for the same reason, looking at the social environment. It could be a resource for going back in time to do developmental origins of health and disease research, or even critical windows research. Maybe it matters if you're exposed to this chemical when you're 2, but it's much less toxic when you're 7 at the same dose. You could start to look at that if we had such a map, or you could at least see whether or not that signal looks like it's likely to be occurring. And gene-environment interactions. I'm a pediatrician. I once had a conversation with the director of the National Human Genome Project, or National Human Genome Research Institute, and I asked him why there are no good studies of gene-environment interaction. And his response was to tell Congress to fund the All of Us study. And I was being polite, but, you know, if you enroll people when they're 30, 40, or 50 and older, and you think you're studying gene-environment interaction, you're ignoring decades of life to begin with in which gene-environment interactions occurred. And I think gene-environment interactions probably are most important in children because that's when development happens. So I don't think the All of Us study is going to be a very good resource for gene-environment interactions. I think if you want to study them, you got to start with kids. And I think if we could go back in time, we could start the study gene-environment interactions. We could even do it in the All of Us study if we could go back in time, but we have to figure out how to go back in time. Because most of the important gene-environment interactions, I believe, occur during childhood. It would also be a resource for predictive toxicology. In fact, I think if we had a Human Exposome Project, it would be boom times for predictive toxicology, because like any omic science, we're going to have thousands and thousands of false positives that we got to weed through. And the fastest way to weed through those will be through predictive toxicology. So it's going to have a major role in actually trying to figure out what's real and what's not. So I think this will actually be a big boost to predictive toxicology. But in each case, the work becomes more relevant to real world scenarios because we want to make sure that the research we do actually applies in actual living populations. Other benefits of investing in an exposome map, clinical usage. I give a whole talk called Environment is Medicine. There's going to be a symposium if you come to the P30 meeting in Kentucky later this month that I'm leading on Environment is Medicine. But basically, I think we tend not to think about people with a disease when we do environmental health research. It makes sense. We're all in public health. We want to prevent disease. But our society has gotten a lot less healthy over the last 30 years, and there's millions of people with chronic illnesses such as diabetes, and we don't actually look at what these environmental factors are doing to their disease. We're trying to prevent disease, and I'm all for that. I'm not arguing against it, but I think we tend to leave behind people who already have the disease. And what I mean by that is if I have type 2 diabetes and I'm exposed to microplastics at very high doses, maybe my dose of insulin is higher than it could be if I wasn't exposed. And by simply reducing my exposure, I might not actually need that higher dose of insulin. And that kind of research, to my knowledge, is not being done. And I really hope that in the future, folks start to do that. I'm towards the end of my career, so I'm sort of dumping it on the younger people in the room. I think that's a big area that we could leverage. Environment is medicine. People talk about how diet is medicine. And I believe that you can reverse type 2 diabetes with diet and exercise. You can also potentially reverse it if you reduce some of your toxic exposures. And then there's commercial uses. Health span and longevity are here to stay. I think, rather than argue that that's primarily for rich people, and I think there's a good argument to be made for that, we should also understand that maybe we can leverage some of that information. So people are very interested in how to stay healthy as opposed to waiting till they get sick to get a disease. So NIH is kind of focused on developing drugs, which, intuitively means when you get sick, we'll do this for you. In the meantime, just wait. People don't want that anymore. They want to stay healthy today. So it's much closer to public health goals. And I think we should probably leverage that in partnerships and actually start to understand how there may be commercial applications to exposomics that actually may be very, very useful, whether it's in real estate or urban planning or diagnostic tools or apps that tell you about exposures from consumer products. The commercial usage, quite honestly, is probably what the government is going to use to decide whether or not to fund a human exposome project. So I don't think we can push it aside if we want it to happen. In fact, Google, Zillow, and other companies already use geospatial maps commercially. Most of genomics is the idea, can we drug that? I hate that phrase, but I've heard that many, many times at meetings when research findings get presented, like, can we drug that? Can we create a drug from it? It's possible maybe we could do that with exposomics. Phone apps, I think, could be very, very useful in exposomics. And as I said, predictive toxicology, I think, will be a benefit because it will be the driver of application. So I think the benefit to those of us in the room is that it could be a way to reconstruct the past, which has been a black box for many, many years. We have these signals like DOHAD that actually maybe our past matters, but we don't have a good way to go back in time. And to do human research today on somebody with Alzheimer's, if you can't go back in time some way, you're going to have a hard time answering that study question. So there's benefits to DOHAD, there's benefits to genetics, as I said, and I think there is benefits to public health. We can prevent disease better if we know when to do public health interventions. Maybe it matters to actually do interventions in teenage years. We can also predict the spatial temporal patterns of diseases and whether they co-locate with different environmental exposures. And we can understand the role of exposomics in health disparities, which are clearly spatial because zip code predicts mortality. So one of my favorite science fiction writers is William Gibson. He wrote a novel called Neuromancer. People say it was the movie The Matrix is loosely based on Neuromancer, pretty loosely based. It's a great novel, but he has a quote in there that the future is already here. It's just not very evenly distributed. So if you think about what he's saying that all the tools we need to develop something like the Human Exposome Map already exists, whether it's AI or whether it's actually the existence of these maps that are currently siloed, but they're not being brought together. A lot of technology is very siloed and not being brought together. And that's really what he meant by that. We don't need a game-changing new technology to start. We just need a program that will organize the data and track what new data is generated. We need a big picture data architecture, as Marlon Miranda would say, that collates and integrates existing resources and builds a coalition of process-led researchers dedicated to exposome research. And so these are all the people who contributed to that. As I said, I'm old, so I've been around, so I've had lots of conversations with people about exposomics, and I probably left a couple people off, but all these people have played a role in helping develop the talk that I just gave. I put a shameful plug for my blog at the end there as well.
Thank you for a wonderful talk. And this is an opportunity for questions. And I know Aisha had already one posted on the Zoom. Aisha, want to speak up, or otherwise I need to dig for it. But otherwise, if there's any questions in the room, Megan?
Well, I thought I should go first.
Is she ready to go?
Hi. Well, it wasn't really a question. Thank you so much for your talk. I'm a preventive medicine physician in occupational medicine, and I direct the occupational medicine residency program based out of our department. And I really connected when you framed the exposome and like the practical application of zip code as risk factor that could be incorporated into someone's health evaluation. And I would love to hear your talk on environment as medicine. So if it's okay, at some point we may follow up with you and see if maybe we could get you to come and talk to our residents. We could maybe do something jointly with our preventive medicine residents, because I think there is so much interest in environmental medicine right now. And I would just love to hear your additional thoughts on that. But thank you so much for putting that in there. I think that it's very valuable to think about how we can apply the exposome on an everyday kind of basis, and it can lead to further identifying opportunities to prevent not just the development of disease, but also, as you alluded at the very end, kind of how disease presents itself by mitigating exposures that could exacerbate those conditions, like we have done with asthma, but for other things. So thank you so much.
Thank you. Asthma is a great example. It's probably the one disease the medical field accepts as being environmental, and I think we could use it as a model. That's great. That's a great example. Thank you.
Great talk. First of all, thank you for the shout out. As a former comparative lit person myself, I appreciated your Borges reference. Exposomics is really compelling, right? It's this wonderful sort of grand imagining of how we can understand things. But what we also know is that all of these data sets are biased. And sometimes they have bias in ways that's really uncomfortable, right? There are sort of structural exclusions of groups as part of this. And whatever the promise of AI, we also recognize that it doesn't tend to help us overcome these biases. It tends to entrench them, maybe even exacerbate them. So as we move forward, what is your thinking about how to try to overcome this challenge that we have with all of our data sets?
That's a great question. So yeah, garbage in, garbage out just gets magnified the bigger the garbage. I think in parallel, we do need a study that is designed to minimize bias. The thought I had about the issue of exposomics is from conception to death. I would like to benefit a little bit from exposomics before I die. And one way that we could design a study would be sort of a what's called you've probably heard of this, accelerated longitudinal design. So rather than a cross-section of people at different age groups that you only measure once, which is what I believe EHE's design is, you follow them. So you enroll women in pregnancy, you enroll infants, you enroll school-age children, you enroll teenagers, you enroll young adults, middle-aged adults, older adults, all in the same calendar year, and then you follow them. And then you kind of connect the dots over time because they will cross over to different life stages. And there are statistical methods to do that. I think that would be the ideal design for a human exposome project so that we could get some answers before everybody in this room dies. Because, that Alzheimer's disease study kind of hits home because there's not a person in this room, I don't care how young you are, that could do that study prospectively. You will die before that study's over. So if you want those answers, we have to come up with ways to get around time. So, and longevity, I think, isn't going to get us there.
You still have your question? Yeah, go ahead.
Sure.
Yeah.
So, I guess I'm not entirely understanding how this is better than what we already currently have. If we already know that zip code is the primary indicator of health outcome, how is exposomics going to actually improve that? Because I see it more as being used in precision medicine, which is going to be used there's no equity built into that. I think it's going to be far more inequitable and going to be used to people from having insurance and things of that nature. So I guess how does this actually translate to like the that?
Yeah, that's a great question. I think part of the answer is that we know zip code predicts health. Zip code is a pretty crude measure of place too. I mean, it encompasses lots of square miles, and in rural areas, it's even bigger than in urban areas. And yet it predicts health, even though it's really, really crude. So part of the argument is if we dug deeper, we could get a deeper understanding. We went beyond zip code to home address and to understand neighborhoods. We could understand why health disparities show such strong geospatial patterns. And maybe we'll start to do something about it. Right now, it's ignored. I mean, physicians, I'm probably in the top 99.99% of physicians in understanding the role that zip code plays in health. I think the vast majority of us have no knowledge of that and don't think of that and never even look at someone's address and say, there's a lot of diabetes in this, address area. Maybe I should screen for that. To my knowledge, that's not being done. And I think if we promote exposomics, we have to think of clinicians. And I think one way to get them interested is to show how it's affecting their patients. And so if, every physician, unfortunately, probably has patients with type 2 diabetes because it's really, really common. In fact, I'm a pediatrician. I was taught in the 1990s when I was in training that type 2 diabetes was an adult disease. And I never saw it until roughly 2012. Never, not even once. And it blew my mind when I started seeing children with type 2 diabetes. So the world's changing. And a lot of this, the interest in exposomics for me is, our genes didn't change over the last 50, 60 years. Our environment did. And something's happening and we're getting sicker. And we're not addressing it when we just focus on, in genomics. And I think I'm meandering away from your question. I realize that as I go on, but I think we have to be cognizant that health disparities are geospatial. And I think we need to factor that in when we design a human exposome project. And a lot of this data exists and a lot of this data is biased, but we got to start somewhere. I think to me, there's this inertia that prevents us from doing this that I hope we overcome soon. We're going to make mistakes. Hopefully we'll, recognize those mistakes and correct them. But the current situation, nothing happens. And I think no one uses zip code. Nobody uses address right now clinically. They should, but nobody does. Did I address your question?
Well, we think nobody uses the zip code, but maybe insurers already do.
Oh, insurers probably do, but healthcare providers largely do not.
So I think also driving the research or the movement into the right hands, leading it forward, might and recognizing that otherwise others are going to get a run with it for their own purpose.
And Fenna and I had a discussion about ethics and exposomics, and I think that's a great topic because once insurance companies see that addresses are associated with higher rates of disease, how are they going to react to that?
Well, they already know. That's why.
So part of the reason why we need this research is we could actually uncover those patterns. If people are being denied insurance based on their address, these sorts of maps could show that. And so they would actually replicate what the insurance companies are probably doing. I mean, the truth is the Exposome Project, that's why I like the quote that Wayne Gibson has, the future's already here, it's just not evenly distributed. Google knows where everybody in this room is right now. I guarantee it. Because they know, you probably aren't one of the people that actually turned off their ability to track you. Maybe you are. So maybe there's a couple of them, but they know where you are. They know even if you're indoors because they can tell by the signal on your cell phone whether or not you're indoors or outdoors. So This information exists. It's just not being used for public health.
I agree. I know this wasn't really a moments forward talk, but a lot of people, when they associate exposure and molecular change, it's sort of this wedding business. And we'll sort of do this in aggregate and GWAS style analysis. I was wondering if you had any thoughts on how that algorithmic approach would be.
Yeah. So the current approach to multi-omics is to look at biomarkers, some of which, like DNA sequence, don't change over time, and some of which, like epigenetic marks, metabolomics, or microRNAs and mRNAs are very time varying. I can tell you with those whether or not you have inflammation, even chronic inflammation. What those studies don't tell you is why. Exposomics is the why. So if I have chronic inflammation, exposomics has the potential of telling me what's causing it. Multi-omics without exposomics does not.
Any other questions? Online? Any other questions? Okay, we are on time, so in that case, I won't probe, and we would like to thank you very much. Yeah. And if there are any follow-up questions, feel free to reach out. There is a blog to follow, etc. And we will definitely connect you with Aisha also for the medical TV. So, thank you. Thanks everybody for coming.